All right, so we're talking about the grant, and we got numerous distinct messages that we should talk about this. And so the thing is I just got an email with somebody that had the 200. We just worked out that the RBD, the R receptor binding domain is critical, 200 amino acids. And I just got an email from some company that makes 214, exactly the same as encoded, I guess, by the vaccine, even put the same leader sequence on. And then they said that they used that to stimulate, I guess, through APCs, maybe even the B cell, which would be perfect to focus the antigen presented to T cells. Now, we know T cells are involved, and if T cells are involved, they must get involved through APCs. I mean, they don't bind the protein that the vaccine makes. If T cells are involved, they are involved because something processes those proteins. And then MHC gets involved because depending on your MHC, you process different components. Actually, we could look that up and
Hmm.
make a T cell tetramer and definitely a CD8 tetramer because they're even easier to make. But they use that peptide, I guess, fed it to an APC, maybe the B cell even, which, by the way, virus specific B cell or RDB specific B cell can focus it very well and bring it in, and then it can use that to present on class 1, class 2 MHC. Must do that, and then they got CD8 T cells that produce interferon gamma, CTLs, I suppose, and CD4s that also produce interferon gamma, so TH1s. So everything is set for that. And then we thought last night, did we already think of it? Because we talked about the APCs, B cells, I went through B cells can do three things. They can produce antibodies. Okay, that's no. That's what everybody focuses on. But they can produce cytokines. Well, that we were kind of good on because we got interferon and maybe IL-6. And they can act as APCs. And we did that because we studied, what was it, John Lang's paper as MZPs as potent antigen presenting cells. Oh, and by the way, this does occur in the germinal center because there's somatic hypermutation. And then you got your memory, high affinity ones that go on to become plasmablasts, low affinity, a big broad spectrum, but they're there for a reason and they produce memory cells. But that gives you a fertile soil to begin with when the variant comes along. So that all that thinking is, what we know about, and that's why we're talking about it. That sounds like it's simple, but it's beyond. We're talking about the grant being significant, obvious approach. We can do anything. So now the innovation. What's innovative? The innovative thing is this whole system. The TB collaboration, the B cells, the APC. And most people don't think of that. They think of dendritic cells or something like that. But we're going to propose that the B cells are the main APC, and they focus the antigen, and probably they focus it. Maybe even they may even if you want to get to some sub aim, aim 3B, is they focus combination material spike, not spike RGB somehow complex with BNP, and that's and there's a BNP T cell response too, and that may or may not be related to interferon. So those are the parameters that we have. It's sort of diffuse because look at this. We're going all the way from immunoglobulin response to T cell, and we have immunoglobulin data. We we have T cell data too. We could look at the immunoglobulin is interesting because the repertoire, we know it's expanded is 330 VH heavy chain V330 and V3 I think 56 or something like that. So those B regions are expanded in B cells. So it's sort of already known that is. But what about the T cell repertoire? Is that also expanded? I mean, it should be. One thing interesting. I know this sounds strange, but I started out studying T cells. I'm very comfortable with the alpha beta chain or gamma delta, but I like that best. But I mean, I know about the alpha beta chain T cell, CD4. So if you give me an alpha beta region, want to make a combo in, I made the T cell receptor transgenic mice to study autoimmunity. So, most people just stick with one thing, like immunoglobulin. And it's so obvious that spike protein or RDB protein antibody is critical to the RDB doing that people don't even bother to go beyond that. But we could definitely go beyond that to B cells as APCs, and then we can even talk about the T cell repertoire. So and then for those patients who don't have a long immune response, the patients have a low response. They're older patients. They either have senescent B cells or senescent T cells. We didn't look at T cell senescence. But one thing that all of them had was this new big population of CTLs. Remember, I looked at perforin granzyme B, and there's CD3 and CD8, big population in all these patients. So CTLs are somehow not somehow, but as everybody said, about 100 years ago, that CTLs clear up the virus. They kill the cells that the virus is in. So we're not saying anything that nobody thought of, but they don't put it together as B cells as APCs. I think that's a novel angle. It's not novel, but we thought of it before is because Schlomchek and what's her name? Yeah, Schlomchek. He wrote that paper. I remember reviewing it 10 years, 15 years ago, that the B cell is the APC that processes the autoantigen. He had some data. That's the thing that went to the germinal center, and we have data on that. So, and interferon plays a role in that, and transport. Interferon plays a role in this virus. So with all this, I think this lays the groundwork that we are uniquely qualified to answer. Right? What do you think?
I don't know. I just don't know the novelty here.
Putting it all together, we're not just studying the antibody, the receptor binding domain, like nuisance five. Trying to squeeze that and look at how long they
Yeah,
last.
hard to say until we get the data. That's the thing. Hard to say it right now.
Luckily, the most of the data is B and T. Right? Remember we sort of.
So we don't have data for T cell from FACS. We don't.
Well, that's a bit of a problem, but.
Right, so we have to use the transcriptome we have.
Yeah. We didn't do any T cell at all?
No. But the T cell, we didn't characterize it.
What do you mean cell population?
That's Sharon said. He just T cell, B cell, natural killer cell, dendritic cell, not anything specific.
Oh. Can you show me that?
From here, you could see this is barbecue 31, and they do have more CD86 compared to the total. So NP,
who does?
NP positive, spike positive. So,
yeah.
Right, so they do have more CD86. You see this is higher.
Yeah.
This is double negative. And then this is naive. So you can see they're both higher than the.
What's the, double negative is this whole group, right?
Right, this is double negative.
So the CD86 and double negative is higher.
Right, and this
now,
is naive.
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 And right. Yeah, I didn't realize that. And so CD86 is higher there, and it's even higher in the spike.
Right. But yeah, so here memory, I may look at memory further with the same thing.
Memory, I think, is good because
Right, that's
memory
right. So,
nuisance slide squirt.
right, because I think we kind of skip memory. I say we're never interested in memory, but now we are. I am. So, but I still want to know what's the difference between this atypical versus the classical memory.
Yeah, and you know, one thing is the memory does go up,
It does go up. That's right.
you know, relative
Yeah.
to the goal.
Right. That's right. So it does go up.
Up here, there's nothing, but you get a specific one.
Right. That's
There is
right.
no memory.
Right.
So
So
those memory B cells might be the ones.
yeah, this is the critical one to
Right,
protect us with the variant. Yeah.
right. Those might be the
Mhm.
critical ones.
Right.
There is. Those are the APCs,
Right. That's right.
as well as the ones that will have a low
Right.
affinity but high, but more
Right.
malleable. They'll get sucked into the new germinal center.
Right. That's
But
right.
see, those memory B
Mhm.
cells right there.
Right.
So I don't know if they have higher CD86. They probably do, though.
So we'll see. But I think they're ready to, I think the function of those memories maybe is to make the antibody more important than to stimulate T cell.
One thing I was noticing, now it makes sense to me, is the naive in the spike was so low. It's it uses
Yeah,
enough.
right. So one thing is I also realized the reason is that this is the surface membrane on the surface, right? So for this antigen to okay, so here now we realize, you say spike, why we are so prone to make antibody against this? And the reason is this thing is on the surface outside. So they use this thing to get into the cell, but then because of that, our immune system also easiest to detect this thing. So
Well, no, wait, they get into, they don't go necessarily.
right, they use this thing to get into the cell, right?
Yeah,
So
yeah,
their advantage, the spike, the advantage for the spike for the virus is also the disadvantage. And that's why the immune system can recognize this.
Yeah, but it doesn't get to immune cells with that. It gets into lungs.
No, but it's outside, whereas the NP is inside that core. They don't see it. The immune system don't see it. So, and I think that's why this is more activated, that you get a lot more DN than this, because this thing for the B cell to see this thing, it's harder than this.
Could be. It goes, if it all gets, yeah. Maybe focusing on DNP isn't the right thing. Nobody likes it anyway.
I still believe it has a very important role.
I know, but
perhaps that's the thing. If we can figure that out, then nobody has it. That's one thing, because everybody has this, but nobody knows what this thing does. But to me, this thing is so important because it definitely, this thing will stimulate the TLR7. And the human B cell specifically, even only two receptor, TR7, TR9. So there gotta be a very important reason for that. And that's why I keep on saying the immune system won't waste it for something to make such a good interferon response to this. It gotta have a very important reason. I don't think this thing signaled through the TLR. This really is the germinal center response.
Yeah, maybe the FC, okay, through FC binding, the whole virus gets yanked into B cells.
Mm,
Could the whole body, it binds to the
mm,
MP, it binds to
yeah.
the spike, and then it's
Mm,
and the FC yanks the whole virus in the B cells, and they act as APCs both for spike and BNP. I
mm,
guess I guess we could
yeah.
I guess we could look at that because if just like we do with the spike,
Mm,
these guys pulse with the receptor binding domain. Or
mm.
no, oh wait a minute. We're studying the vaccine.
Yeah, the vaccine doesn't have the, but it could be the infected convalescent. Oh yeah, well, I think we need to prepare for the Delta variant. So I think there will be more newly infected.
Yeah, if the Delta variant comes along, then we can start talk about what happens in natural.
Yeah, so I think it will. The way things are, Alabamian are 50% refuse to get vaccinated.
Well, that's not much. I mean, in the whole country, maybe it's a
Mm.
75% anyway. 25% are holdouts here and there. And what I'm wondering about is this breaks down into, do we want to study vaccinated people, which
We
would be,
just put in our mind, we cannot decide exactly what we would do yet. I don't want to give up on this thing. That's the thing. I
right,
think I understand this a lot more,
right.
but I understand this less. But it's just I cannot get rid of the thought that this thing must be so important. It might be even more important than this. On Earth, the immune system devoted entirely for this thing for interferon is devoted for this. I just cannot believe. I just can't imagine that there's no one very powerful reason for that.
Yeah, but from a grant's point of view.
No, I know. So unless we can figure out why, of course I can give up on just focusing on this. But to me, everybody's focusing on this. So, but if we can figure out why this is so important, if we cannot, forget it. I would say forget it too. But if we can figure it out, that's the advantage we have, because we developed this through our understanding of lupus that nobody has.
But in one case, we're studying, of course, and that would only apply to the natural infection, which
Right,
is
only natural. So then we say because of this, the vaccine response is worrisome.
right.
So that's the reason. That's the significance and novelty here.
If we go with the
Mm,
if we go with the same idea that we've been talking about,
mm,
if we include that, it
mm,
seems like we have to have, I mean, I'm pretty sure it means
mm,
we have to study two systems, two sets of T cells and B cells. One B cell reacting with spike, and we
mm,
know that they don't overreact. So the other B cell are BNP.
right,
The spike ones we're saying are APC. So that we know
right,
an APC carrying cell.
right.
So we can work
Yeah,
off.
so this is the antibody forming B cell. That is to make antibody. These B cells, there's only one purpose for them. That is to make B cell. It make antibody. There's
No,
only
no, no, no. They could be antigen carrying cells too for the in the case of
oh
the
yeah, that's right.
they
Could
can
be,
carry spike
yeah.
antigen.
I think the transcriptome will tell us. That's why the transcriptome is critical. The prediction, of course, would be this is the type 1 ISG high, right? If
Yeah.
that's the case, then we have a story. If any that this is the ISG positive, then forget about this, I would say. That make it simpler. We just focusing on the spike positive B cell, right? Life will be
The other thing,
easier,
the natural people with natural infection,
right?
are they different from people with vaccine? We do have a large
No, I
number.
know. So we have my B cell will be very good because it's really so
Right.
specific.
Right, we have data on,
Right,
a lot of data on naturally infected.
right. So
Remember,
me
we
and
focus on that.
yeah, right.
And then
Me
we
and
have data
David,
on
right,
vaccinated.
right. So we'll see. Right. What's the difference? It's not B cell receptor. That's not it. Yeah.
I think the natural infected ones don't get a lot of, and they don't get a lot of CD8s, I think. I'm not sure. Did we look at that? Or no,
Uh,
vaccinated people, they don't get this big CD8 T cell response.
maybe not.
They
No, I
probably
don't
have a
think
different
so.
B cell. I
Right.
mean, it's CD4.
Well, but who know? Maybe you during the recall response, maybe,
You mean of the vaccinated?
but vaccinated, the recall response, maybe.
You mean if you get infected again?
Right. If we get infected, maybe the vaccine will enhance the CD8 response.
Well, I think that's right. I think that, but that's a second part of its protection. But
Right.
what I mean to say is, in
Right.
one case, we're studying natural infection, which you have both arms going.
Right. That's
In the
right.
other case, if we study
Right.
vaccine, we just have the lower
Yeah.
arm.
Right.
And activate the upper arm, the BNP arm.
Right. Yeah. Yeah.
So maybe that's an angle for the grant, to study the
Mm.
differences between natural infection and
Mm.
do APC B cells have a role in natural vaccine or
Mm.
or natural infection? We know they have a vaccine, but the question is, are they involved with natural infections?
Mm.
And do they and does a natural infection evoke a longer, better response
Mm.
because of now you got the DNP APCs?
Mm.
And the same thing, we could do the same thing. We could take the VNP protein, you got that, and pulse sort the B cells or pulse B cells and show that they activate. This same paper,
Mm.
you pulse the B cells from a person with vaccine, with natural infection versus another one with either the spike, like the paper did, look at T cell CD8, CD4, or you can do the BNP. You can use that to pulse it. And again, you look at the same T cell response and see if, I would say that the T cell response should be very low in people who have just been vaccinated. The BNP response, not the BNP response, the T cell response to a pulse BNP.
Mm.
Of course, we know anti-BNP is low, but so the B cells aren't even there.
Mm.
So maybe something like that. I don't know if anybody's interested in that, whether they're just interested in the vaccine response. But, the grant was based. I think the pathways for natural infection versus vaccine don't have to necessarily be so different. They just have to utilize different epitopes for being B cells and then the T cells.
Right.
But I do think it'd be good to tie in the T cells.
Mm.
And then because the grant almost writes itself, because now everything's there. It's a rubber stamp, APC B cell and then CD8, CD4 T cell response, and you got two different pathways.
Mm.
By the time the experiments you do, you know what we do. We got the tetramer, we got the single cell, we got the transcriptomes, we got the TCR and BCR repertoire. So we got everything. And it's a matter of answering those questions. The question, here's what I think the hypothesis is. We think like this. The combined response to BNP plus spike, the combined response of both of those will give either a better response, a higher response, or a more persistent response, or a better recall response. But in other words, the response is more protective. It's either last longer, or you get a recall, or the more diversity, or it'll last longer and be more available for the variants that come along. So I think that kind of thing is significance. We could propose that. Who's going to say, "Oh yeah, no, that's not right"? Because nobody probably. I hate to say nobody, but we think at kind of a level that's universal and pulls anything in. I think most people are so focused down on one thing. So this makes a nice enriched grant.
All right.
The hypothesis is to get a long-lasting and beneficial response to variants you really need the VNP tied in it because that's an invariant. You know, so you need an invariant plus a variant one. How's that?
All right, for now, we can settle for that for now.
For now, I can dictate the first page.
Oh, all right.